Hello, Goenkans. Hope you are doing good. We were going through crop production. We were done with two steps of crop production. One was preparation of soil, that involves ploughing and leveling, and second was manuring, which included manures and fertilizers. Today, we have to go through sowing. It is chapter 1 of your textbook, page number 6. Fine, let's go through sowing. What is sowing? The process of putting seeds into the soil is called sowing. You're putting seeds simply into the ploughed soil, leveled soil, it is called sowing. Fine? Because the first step is ploughing and leveling. First of all, we have to plough soil, then we have to level it, then we add fertilizers, manures, and then we put seeds. And the process of putting seeds into the soil is called sowing. Before sowing the seeds, the following precautions must be taken. What are the precautions which we take before sowing seeds? And this we are not doing on small scale. If you are doing it in your kitchen garden, you don't follow such things. But if someone is doing it on a large scale, farmers do it on a large scale, 60% population of India is involved with agriculture, isn't it? So they need to take precautions, they need to follow proper steps. So the precautions which one must take before sowing are: seeds must be of good quality, healthy, disease resistant and pest free. We cannot take seeds which are not healthy, which are from a diseased plant. We have to make a wise choice, isn't it? We have to take seeds of good quality. We have to check their quality. We have to see if we are taking it from a plant. Is that plant healthy? Are these seeds disease resistant? Are they pest free? If pests have attacked those seeds, we cannot sow them. We will not get good variety of crops. We will not get good yield. So we need to check seeds first. So the first precaution is seeds must be of good quality, healthy, disease resistant and pest free. Our second precaution is seeds must be sterilized by fungicides, antibiotics, etc. We have to sterilize seeds first. Sterilization means making something free of microbes, germs. Nowadays, you might have heard this on news channels. Sterilization is going on everywhere. What does this sterilization mean? Sterilization means making surfaces free from germs. So remember this term, sterilized. Actually, sterile has two meanings. One is opposite of fertile, fine. Fertile, sterile we call. Fertile is, what does fertile mean? For example, if we talk about fertile land, which gives us good yield and all. And sterile means barren, fine. In Urdu we call it banjar, fine. So that is one meaning. Another meaning of sterile is making something free of microbes. So we have to sterilization we have to do. We have to make them sterile. How we make seeds sterile? We put fungicides on them, antibiotics on them. Now what does fungicide mean? Fungus pertains to fungus, and side, C-I-D-E. Side, wherever you find in biology, that means to kill. Fungicides, pesticides, fine? Bactericides, something which kills them. Bacteria, fungi, etc. We call them sides. So we have to put fungicides on them, then we can put antibiotics on seeds. Now the question is, what are antibiotics? Anti, opposite. Fine. So anti, wherever you find, it means opposite. Antibiotic. Biotic means it pertains to life. So here we can say, I can give you definition of antibiotics. Any substance that can destroy or inhibit the growth of microorganisms like bacteria, etc. Substance which can destroy, fine, or inhibit. Inhibit means which can curb the growth of microorganisms. We call them antibiotics. So we have to put fungicides, antibiotics on these seeds so that they get sterilized. Fine. Next is there must be proper spacing between the seeds to avoid competition for space, oxygen, water, nutrients, and light during the germination of seeds. Spacing should be proper. We cannot put 2 3 seeds in one hole. No, that is not the way. There should be proper spacing because they will grow seeds. They turn into seedlings. They should have proper space. Then there should not be competition for space, oxygen, water, nutrients, etc. So spacing should be proper. Then seeds should be sown at appropriate depth. Again, that's very important. Depth is very important. If seeds are sown too deep, they will fail to germinate. Fine, they will not germinate. If you are interested in gardening, you should know about these things. Fine, so they will fail to germinate if you're putting them very deep. And if you put seeds close to the soil surface, they can be easily eaten by birds and other animals. So we have to maintain proper depth, appropriate depth. So seeds should be sown at appropriate depth. Seeds can be sown by any of these methods. There are methods of sowing seeds. First one is broadcasting, then there are traditional tools, then there is seed drill, and then there is transplantation. First is broadcasting. This is a very simple and traditional method. This is a traditional method where seeds are scattered on the field by hand. Simply you are taking a handful of seeds and just scattering it, throwing it, fine. But there are disadvantages related to it. The disadvantage of this method is that the seeds may not get sown at proper distances and at proper depths, resulting in wastage and reduced crop yield. This is quite obvious. If you are just scattering them by your hand, obviously proper depth will not be maintained, isn't it? And that will definitely affect the crop yield. Second is traditional tools. The traditional tool comprises of a funnel connected to 2 to 3 tubes that have sharp ends. You can look at the picture and just try to observe what I am saying. The traditional tool comprises of a funnel. Funnel-like structure is there. You know what is funnel, isn't it? We have seen this funnel in our lab as well. So funnel is a conical structure, isn't it? So what happens in this traditional tool, there is a funnel that's connected to 2 to 3 tubes that have sharp ends, fine. And seeds are put in the funnel, they travel into the tubes and then penetrate into the soil. And what they do is they place seeds at proper depths. Proper depth is maintained. This tool also ensures the seeds are spaced correctly. Fine. But there is a disadvantage. It requires more time, more labor. See, manpower is involved, then animals are involved in it. Although it is better than broadcasting, but then again it requires more time and labor. Fine, you can observe it. See, I will try to show you with this. What is there? There is a funnel-like structure. Here is a funnel-like structure. And then there are these sharp tubes. What they do is they put the seeds at appropriate depths. I hope I am clear. Please go through your book, read thoroughly, and answer few questions. Write them on your fair biology notebook. Question number, you can attempt question number 3, question number 4, and question number 5. Fine. Tomorrow we will take seed drill and transplantation. Thank you, Goenkans. Stay indoor. Stay safe. Bye-bye.